Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show. Today, bringing another really cool guest involved in science and technology and healthcare, creating a better tomorrow for many people out there. Today on the show, we have the honor of being joined by Edmund Benami, who is co-founder and chief executive officer of a company called NeuraLight. They are a really pioneering health tech company. They're focused on applying advanced computer vision, deep learning techniques to accelerate and improve drug development as well as patient monitoring in various neurological diseases. And their mission is ultimately to develop these really objective and sensitive biomarkers in neurology by extracting so-called oculometric markers. These are measures of various eye movements from standard webcams, smartphones, ultimately providing a really rich set of digital biologic endpoints for, again, neurological drug development, patient care. Eddie is a repeat entrepreneur. He's also an award-winning mathematician with over 20 years of experience designing these novel artificial intelligence algorithms, building world-class teams to take on some really interesting technological challenges in signal processing and computer vision. Eddie has both a bachelor's degree and a master's in mathematics from Ben-Gurion University of the Negev, and we're excited to have him. And a lot of topics we don't normally get into, but really interested in hearing and learning what's going on. Edmund Benami, thank you so much for taking time to come on the show.
Thank you so much for having me, Ira. It's very, very nice of you to spend some time with me.
I'm sure I'm really looking for this conversation. And again, as we don't normally get into these themes, I'm going to learn a lot from you. Would love to start off at the beginning. And you know, I have a life sciences background. I know very little about mathematics, but I could tell a little bit taking a look at your thesis and some really interesting work that you were doing in this area of reconstruction and inverse problems, and in your case, looking at algebra and creating geometry and topology out of it, that there are some very interesting connections, let's just say, to where we're going to be talking about eye movements and, you know, coming up with, you know, some understanding in sort of the hidden biology, let's say, of various diseases. And I would love if you could walk us through a little bit of the early days and what your thesis work is all about, because I think it's directly applicable to where we're going.
So first of all, I think it's so nice to know that someone is looking at those papers. You know, usually in the world of math, it's like you're always really happy if more than 4 or 5 people actually looked at what you did. So I think you almost multiplied the amount of people that glanced at it. So thank you so much. So yeah, overall, my background, I was doing geometric group theory, and in general, my interest was always about extracting robust and rigid information from very complex structures, which is kind of the counterpart of extracting insights from like really noisy, high-dimensional data. And that's kind of what machine learning and AI are trying to help us with today. So that was kind of always what I loved doing. I was doing that during my academic time in a more theoretic kind of setting. And in the last 10, 15 years, I am trying to take everything that I like and know and find use cases that are like, for me, I prefer use cases that are related to healthcare. That was always my passion, as someone that disappointed their grandparents by not becoming a doctor. So that's kind of the second best thing I can do.
Absolutely, absolutely. And it's quite interesting, when you're talking about these use cases, and we'll be getting into a lot of what you've constructed there at NeuraLight, but would love to start off just by introducing the audience to the principle of these oculometric markers in general, because there's again quite a few of them, in the sense that we don't think of all the things our eyes do. We blink, our pupils dilate, we gaze, we do the smooth pursuit stuff where we follow things around, and they all they're sort of common things that our eyes do, but they could all reflect a wide range of activity, from autonomic nervous system, dopaminergic system, life and death. I mean, if our pupils don't dilate, that's a problem. Talk about just what oculometric markers are in general, and then we'll take it down the road to what they mean in terms of potential biomarkers and then digital biomarkers of disease.
So first of all, it's a great point. It's I think everyone knows the saying that eyes are the window to the brain, the soul, the brain, however you want to take that. But this actually has a reason. It's the same reason that any of us that comes to be tested by a neurologist will always be asked to look at their nose and then follow their finger. And the reason for all these tests is that on one hand, eye movements are incredibly complex, and they involve most of the regions of the brain, and the neural pathways are complex enough to give a huge amount of information about brain function on one hand. And on the other hand, together with this complexity, it's kind of one of the very few things that we understand well about the brain. And the reason is, incidentally, that you can actually teach monkeys to do things like stare at a point or follow a dot on a screen. So in the last 70 years, there was a huge amount of work on apes that was meant to understand better the neural pathways that are related to each of those movements, like all these single neuron works that started somewhere in the 50s, where people were trying to decode the uses of specific brain functions and what each of them does and how they're used in more complex situations. And thanks to that, like on one hand, you have something that's very complex and gives information about so many parts of the brain, and on the other hand, you understand what it actually means. You can use that to actually gain information about what's happening in there. So as you mentioned before, it turns out that, for instance, when a person is following just a smooth movement, what's called smooth pursuit, and some aspect of that are a very good way to understand whether their cerebellum is actually functioning properly or not. And when someone is looking at specific aspects of when a person does saccadic movements, which is these super fast movements that we use to scan a large area. So we look a little bit here and then jump really fast to a different place. So those are, in those, the basal ganglia are playing a very, very important role. And here I can say in parenthesis that basal ganglia dysfunction is one of the main manifestations of both Parkinson's and other diseases like PSP. So taking all of those together is kind of looking at movements that are related to eyes on one hand, but looking at them as a manifestation of brain function. And in general, measuring brain function is the most interesting thing. And it's cool that you can translate it to looking at very basic things and measuring things like, we're measuring things like response time to something happening on the screen, or how accurately a patient is managing to follow a specific pattern. So, and the interesting part is that we not only understand how each of these measurements are related to specific brain function, but how each of these measurements are related to either very motor manifestation of specific diseases, or purely cognitive manifestation of other diseases. So it's like a
Mm-hmm.
very cool way to kind of have an imaging of function and not imaging of structure, because MRI
Mm-hmm.
is doing very good work in imaging brain structure, but measuring function is way more complex. You can't, like, unless it's
Right.
an ape that does the trial, you cannot really connect the electrodes and easily do measurements inside the brain.
Mm-hmm. Mm-hmm. And could you just take us? I mean, we'll get to what NeuraLight has ultimately put together in a little bit, but there is a long sort of parallel history here in terms of the development of early forms of oculography, from taking photographs of the eye and then going into the sort of the 1940s and 1950s, where this electrooculography first started, where Parkinson's and Huntington's and schizophrenia and thing like that sort of became something that could be tracked. But could you just say a few words about sort of the old school methods of electrooculography that have been developed over the last century, and then we'll drill down on a little bit of what you've been working on.
Sure. So first of all, it's a really exciting subject since, as you would expect, a lot of people realized already about 150 years ago that eyes are an amazing source of information about brain function. So the first works were somewhere in the 19th century. Psychiatrists in Germany were measuring eye movements to try to accurately quantify aspects of psychiatric disorders like schizophrenia.
Mm-hmm.
And at those times, everything was mechanical. It was super complex. They were kind of gluing mirrors to patients' eyes and doing something very manual to measure these movements. Fast forwarding to when there were methods that were using electricity, as you mentioned before. So it was either a coil that was glued to the eye and measured movements, or something that was attached to the face close to the eyes and was measuring electric signals that were meant to move the eyes by, when electric signals that are getting to the muscles that are moving the eyes. In the last 30, 40 years, most modern methods are using things like either infrared cameras that are following eye movements. They're using the fact that the back of the eye is reflecting infrared light much better than everything else, or just video and then computer vision to measure eye movements. I think in general, most of the challenges that eye movement science had in the last 100 and something years was that usually these methods were having problems with generating very accurate measurements. So it's not very surprising that, as you mentioned, I think a few minutes ago, that when a physician is testing someone's pupils with a flashlight, so if they're dead, the pupils are not going to respond. But if you're trying to do something which is way more accurate, then you need to be able to measure very, very small movements and also be able to assess the time when a movement happened in a very accurate way, because in the brain things are happening super fast,
Right.
and the trying to do that meant, for once, that if you want to be accurate, you need to make sure the patient head is not going to move while you do the measurements. So that already makes everything super complex. You also need to make sure that all this very complex machinery is going to all be very aligned and synchronized in the sense of timing, because if you want to see how patients are reacting to some light bulb that's turning on and off, then you need to really synchronize the light bulb with the other machines. And it was always a very, very big technical problem that neuroscientists had, just to deal with the technicalities on one hand, and with the fact that it's simply very uncomfortable and very long and tedious process for the subjects or the patients. So
Mm-hmm.
in the end, the bottom line was that the amount of data that was collected in the last 100 years is not very large, not as large as we would hope. A lot of the data was collected on apes that, had no choice and were just there and being measured. And in a way, what we were trying to do as like our guiding principle was just to take all the science that people found out and just try to make this something that's easily useful without all the challenges that I just mentioned, that, you know, you need to hold the patient's head, and that it's super long, you know, sitting hours in a dark room, and the fact that it's so technical and you need so much knowledge just to use these machines that were being used in the past.
Mm-hmm. Mm-hmm. Yeah, well, that segues very nicely into where we are today and sort of the new era of digital oculometry that you're ushering in here with the computer vision, with the AI. You wrote a very nice review in Frontiers in Digital Health. This is back in July of 2024. It's entitled Advancements in Eye Movement Measurement Technologies for Assessing Neurodegenerative Diseases. And you talk a little bit about the strides in image sensors and computational power and the cloud and everything else. Take us into sort of because I look at NeuraLight right as two things. One, a major technology story and you creating this important platform, and then obviously the validation in different disease states. And we'll get to that. But talk a little bit about sort of the early days of the company, before the stealth, or coming out of stealth, let's say, where you built a lot of the video capture, the signal processing, the AI stuff, of course, the data sets. Talk a little bit about sort of what went into the early work at the company in terms of on the tech front, and then you could sort of describe sort of the initial, what I'll say, product is in terms of digital oculometry as we sit here today. That'd be great, and then we'll start talking about some diseases.
So, as I mentioned before, the main thing that we understood that we need to do, so like when we started thinking about starting NeuraLight, the big thing that we realized is that there is so much understanding of the meaning of each of those eye movements and the measures behind them and the relationship to brain function, and the kind of barrier that keeps that from being useful is simply the friction and the difficulty to collect high quality data in large quantities, because for that you just need it to be something that's easily useful. So we started from a place where the nicest to use tools were something that needed tech capabilities and had much more constraints about how they're being used. So you do need to make sure that people will not move too much, and there were like a lot of constraints regarding how they need to be situated in the room and in front of a special screen and so on. And the first version of our product was just a standard screen of a PC and the camera that we attached to it. Probably both the screen and the camera are cheaper than the ones you're using right now. And the idea was to build something that, on one hand, works and manages to generate data that is very accurate, and on the other hand, to be able to, as quickly as possible, start bringing it into testing both healthy individuals and as many patients as possible. Because it was clear to us that the most important thing is not the technical accuracy of measuring, some technical parameter of the eye, but figuring out how, using a situation where we have a patient, a screen, and a camera, we are extracting information that is both reliable and valuable in the sense of giving actionable information about their brain function, which meant that these are questions that we can answer only once we have a lot of high quality data, both of healthy individuals and patients. So as quickly as possible, we got to a place where we had something that was working, and we started collaborating with very smart neurologists and with really cool clinical centers, because the goal was to just get as much data as we can, since, a very good rule of thumb is that when you try to solve a very specific question, you're probably going to find a much better solution than when you try to, solve everything in one shot. So I don't know how much our listeners know, but in general, there are a huge amount of uses for just, following eye movements in the sense that figuring out where a person is looking on a screen, all the way from, I don't know, questions around engagement with ads to gaming to VR. And there is like a huge amount of things that you would want to do with that. And when you try to solve the problem in general of accurately understanding where a patient looks, it's a really complex problem. Like, so many smart people have been working on it for decades. And what we said was, we're not trying to do that. We are trying to extract these specific, what we call the oculometric markers, the markers that are relevant for brain function and for specific diseases. We're trying to extract those in a really, really accurate way in a very specific situation where patients are not looking at a YouTube video, they're looking at a dot that's doing a very specific movement on the screen. And once we realized that we're solving a much simpler problem and a much more specific one, then we could also give a much better solution. And the moment we had that, we started collecting very high quality data without asking patients to, sit in a laboratory for 3 hours with their head pinned down to something. And once we managed to collect a way larger data set than anyone has ever looked at of that sort, then we got to a point where we could start honing in on exactly what are the things to measure to give the most value. And on one hand, on the other hand, I should openly say that, unsurprisingly, so many of the cool neuroscientists that have been interested in eye movements and doing this research for many years became very excited to collaborate with us, simply because they said, oh my God, there is such a huge data set. We've never seen so much data in the past before. So it was kind of clear to everyone that now we just need to, do a good job, and we have what it takes. Now we just need to do our part, and we're going to have something that's going to be really valuable, that's going to come out of that.
And some of your, initial sort of clinical partnerships had to do with ALS, with Neurosense Therapeutics, this back in 2022, and they also worked with the CHDI Foundation on Huntington's disease. And I was wondering if you could just talk a little bit about sort of, some of those initial validation, because, again, this for the audience, in my rudimentary understanding, where ALS is more of a complete motor, deterioration of several of the functions, while Huntington's, I think, initially starts with problems with gaze control. And can you just talk a little bit about, sort of what happens in those collaboration? Obviously, nothing confidential, but what's the first thing you work on, in terms of, all right, this is the proper, first biomarker that we should be, because this will tell us, hey, 4 months beforehand, these important milestones, let's just say, in the progression of the disease. Take us into a little bit of what that work entailed.
Of course. So first of all, just to give the context. So as what we are doing is extracting biomarkers that are meant to measure deterioration. So usually there are like two kinds of biomarkers. There are the ones that are used for early diagnosis, and then there is the second kind, which is what we do, or rather what we're focusing on in the near future, and that is assessing deterioration of either a specific disease or a specific function. Yeah. So first of all, that means that the end goal is giving our partners biomarkers that we can tell them that they are measuring function of specific pathways that are relevant to the specific disease they're interested in, and that are much more sensitive to change than anything they have or have used in the past. So for that, we kind of needed to go through the validation that you mentioned. And the way our validation process looks like, and that is, in general, we have these three pillars that are extremely important to understand how we look at our biomarkers. So the first pillar is, as we mentioned before, we are not a black box AI that's looking at lots of signals and hoping to combine them in a way that's gonna show something. We literally know, because of all the neuroscience, what are the specific measurements that are interesting, and our use of AI and computer vision is to make those measurements as robust and accurate as possible. So the first pillar is understanding what we need to measure and what it actually means. The second pillar of validation is showing that these measurements are correlated in data that we're collecting from patients to everything that you would expect. So if you have a disease that has different kinds of manifestations that are related to different kinds of dysfunctions, then you would expect a marker that's measuring cerebellar dysfunction to be correlated with aspects that are symptomatic aspects that are relevant to cerebellar dysfunction, and something that measures basal ganglia dysfunction to be correlated with things that are related to aspects like symptoms that are related to basal ganglia dysfunction. So we have these correlations to the right things that are convincing us and everyone else that we're measuring the right thing. And then there is the progression validation. And usually what happens with our partners is that they're joining at some stage of this process. With Neuroscience, they were our partners from the start, and we went through a lot of the validation of our ALS biomarkers with them. On the other hand, with other partners, they're joining us on working on a specific disease in a place where we're already fully validated, and it's just a commercial partnership where we're giving them services as a biomarker company. With the CHDI, it's more similar to the first kind in the sense that we have results that are related to Huntington's disease, but them together with us are committed to bringing more biomarkers that are going to support drug development as fast as possible. And we're literally working on that together. They were helping us to collect, just recently helping us to collect lots of patient data to accelerate this validation process. And in general, the bottom line is that once we have, for a specific disease, a list of markers that we understand what they mean, we understand how they're related to specific aspects of the disease, and we have multiple clinical trials where we showed that they are much more sensitive to disease progression than the current gold standard, then now you have something that our partners know how to use. They know what this means, they know what it will give them, and they know what to expect when they're using this in the context of a clinical trial.
Yeah, you said something important there. I want to focus in on that because of obviously beyond the earlier diagnosis and the more precise monitoring, clearly many of your clients that are going to be coming online are going to be, drug developers. And when it comes to some of these neurologic conditions, from just, I have a little experience in this space, these trials can go long. I mean, some of them can have to go for years based on what you want to see and what the FDA recommends that in terms of labeling and all that. Say a couple words about that, because this whole principle of de-risking drug development, if digital endpoints of this nature can tell us something years before, so instead of spending $700 million, only have to spend $200 million. That's major. And I want to stress that one a little bit more, because I think it's important, because we don't really think, okay, wow, maybe we think about biomarkers and blood and all that stuff, but maybe there's something here that, hey, tells us a little more, and then obviously a little earlier than waiting for that data to be unblinded 5 years from now or whatever.
So first of all, it's a great point. And I think maybe even it's worth to emphasize for the listeners that didn't have the unfortunate chance to be close to someone that had to go through a process like that. So the main tool, the gold standard for assessing efficacy of a drug today, and it's also the main tool, it's kind of the main tool in general in a lot of those diseases, is questionnaires that are filled by a physician. So the way today we decide whether an experimental treatment or drug is helping Parkinson patients is that we're enrolling lots of those patients into a trial, and the way we measure how they deteriorate throughout the trial in comparison to the patients that were not treated, what we do is like a physician is filling out a list of checkboxes where, for instance, you want to assess their balance. The way it happens is that the physician is pushing them and they're checking how hard they need to push them for them to lose footing. Or if it's an ALS drug trial, then they're asking them a bunch of questions just to give a general feel. One of those questions is like how hard it is from 1 to 4 for the patient to adjust their bed clothes while they're sleeping. And this really crude tool, which is very based on the perception of the patient and the intuition and perception of a physician, this is what is being used to decide whether a drug is working or not. And first of all, of course, it means you cannot even dream of quantifying how much it works, because even just figuring out whether it works is so complex. And this is why drug trials lately are, first of all, just as you said, very long and with lots of patients, because you need to make up for all the subjectivity and the problems with the measurement. And just last year, a very important publication of the Michael J. Fox and a very famous Parkinson specialist came out, where they actually got to the conclusion that with the current length and size of drug trials, we expect a working drug to fail with 95% chance.
Yep.
Not succeed with 95% chance, fail with 95% chance if it's a working drug. And so what happens is that pharma companies and patient associations and foundations that are trying to promote cures are actually working really hard to try to find ways to measure whether a drug works, which means ways to measure how much a patient deteriorates. And here I should say, just to, so that it's clear that this is not meant to say why are people doing these weird things. There is a reason for this shift. So until just a few years ago, in neurology, drugs were mostly symptomatic drugs. And what medicine was looking for, because that was thought to be the solution, was something that the patient will take, I don't know whether it's a pill or an injection, and all the symptoms will kind of remiss or go away. And in that situation, what you needed to see whether a drug works was just something that could compare their situation before they took the pill and after they took the pill. Now we're trying something way more ambitious. We are trying to find medications that will actually slow down the deterioration of the underlying disease itself, maybe even stop it or even reverse it. And then all of a sudden, it's not enough to check whether a person got a shot or a pill and now they feel much better. You're actually trying to see whether their deterioration over time is becoming slower. And that is just something we never tried to do before. And as it happens with humans, the system is working the same way it did before. So we're using tools that were meant for finding symptomatic medications to try to find disease-modifying or neuroprotective medications. And this is why there is such a huge need. And pharma companies and all these organizations are trying to as quickly as possible, find ways that can quantify how fast a patient deteriorates. Because if you want to say, what does it mean that a drug slows down progression, right? You want to say something like, this medication slows deterioration by 30% or 50% or 70%. You can't expect to do that when you barely can say that the patient got better or worse in a very long period of time. And of course, once we have good tools that do that, and this is where we're coming in. So our goal is to be that new gold standard of assessing how much a patient deteriorates. Then, as you said, first of all, we want to make sure that we never miss on drugs that actually work. But we also want to be able to we want to enable pharma companies to do smaller and shorter drug trials so that they can go over all these promising molecules as fast as possible. Like it's kind of mind-blowing that molecules that are being tested today are molecules that were invented about 20 years ago. And you want these loops to be much faster, and you want the process to be much faster and much more reliable. And that is the value that we give our pharma partners. And that's why, by the way, one of the only things that they care about, besides getting convinced that our measurement is expressing what they care about, which is the internal state of a patient, that is just to see how fast and how accurate we can monitor progression.
Mm-hmm. And Eddie, I know that, in reading a little bit about the company, I know you've been focusing in also on Parkinson's disease, and you've made some really interesting findings there in terms of how sensitive your technology is compared to sort of the movement disorder standards, and I'd love to hear about that. But at the same time, it gets me thinking about sort of the personalization component here, because and again, we talked about these on the show. Obviously, we talk a lot about Parkinson's, but many people don't know much about progressive supranuclear palsy and corticobasal degeneration and Lewy body dementia, and all these other subtypes of the two biggies, Alzheimer's and Parkinson's, which are different diseases. I mean, they're not the same. They have different presentations, and I'm assuming that the oculometric dynamics of each of these could be quite different. And just love to hear from you. I'd love to hear what you're doing in Parkinson's and some of the things you're finding out there, but a little bit of your thoughts and learnings about the personalization and how a lot of those lesser known diseases, a lot of people don't know how to diagnose them that well. And even in 2026, talk about that a bit.
So first of all, you're completely right. And so just as there is this huge challenge of figuring out what new drugs are doing to patients, it's very clear that if it's so difficult to assess deterioration, then it actually means that even a given treatment, like if you already give a patient something that works, then understanding how much it affects a patient and whether you need to change treatment or not, that is going to take a huge amount of time, because you need the deterioration to be immense in order to understand what's going on. So we're not only trying to support drug trials in the sense of looking for new therapies and looking at groups of patients, but also understanding better how we can treat specific patients. But just as you said, there is a very big problem of discerning between different diseases. And I think there are even lots of celebrities in the last few years that were misdiagnosed. And this is a very, very big challenge. For us, we realized that, so because our view is that we need to do lots of validation first, and only then we go out and try to collaborate. So we decided to postpone the diagnostic efforts to a later stage, because we felt like we need to focus on one of them. And we felt like our fastest way to impact as many patients as possible would be to help support finding a cure and using given treatments in a better way, which meant that we need to understand how to measure progression. And in the sense of Parkinson's, then it was so, as I mentioned before, the way our validation studies look like is that the last big step, which we usually repeat many times, is figuring out how much more sensitive we are to progression and deterioration than the given gold standards, which incidentally are these questionnaires that we mentioned. And in Parkinson's, we very recently had this huge milestone that we had yet another clinical trial, which was very large and in collaboration with multiple clinical centers. One of them is Rush University, which is one of the leading clinical centers in the world for Parkinson's research. And the huge milestone on our end was that the conclusion of the trial was that we are 10 times more sensitive to progression than the gold standard questionnaires that are used today. And the results were so overwhelming that the paper that was sent to publication a few weeks ago was co-authored with the two creators of the gold standard, with Professor Christopher Getz and Professor Olivier Rascol, who are the two guys that, I'm not sure guys is the right word because, they're luminaries, but the two clinician and neuroscientists that stood behind defining the currently used gold standard of how to assess deterioration of Parkinson's. So in this sense, it was really huge for us. And we feel like besides the scientific and validation achievement, it also made a lot of our partners feel much more comfortable with using us in a larger span of use cases than they did before.
Absolutely. Well, seems like your partners love what you're doing, and so do your investors, and you're being very successful on that front as well. Eddie, I want to ask one other thing, and again, get your perspective here. Very important topics for us on the show in the last couple of years have been aging in place, healthcare in the home, and then this whole theme of sort of the distributed clinical trial, the virtual clinical trial. Love to get your perspective on how you see the technology evolving in terms of taking it and democratizing neurological assessments during some of these clinical trials where you might not have to bring in all the patients to a center and do X, Y, and Z scan, but make it part of sort of this remote clinical development movement that we're seeing in recent years.
So first of all, I love that you guys are talking about this a lot, because I feel like one of the main issues in healthcare in general is that, many times we love talking about the forefront of science, but that's usually something that happens in 3 clinical centers and 5 geniuses that can help you get there. And then there is all the rest of us. And in this sense, and these are things that are coming up in your show many times about how the fact that we can use AI is something that is very helpful in at least supporting. We never talk about substituting a physician, but at least supporting some of their actions in a way in which we don't need someone that is like a superstar, that you have only 5 people like that on Earth, and at least some of what they do can be done, with a phone at home. I think that in this sense, I totally agree that this is maybe the biggest challenge of using AI the right way in the next, 20, 30 years. And I'm sure you're not going to be surprised that the reason that from the beginning it was very important for us not to be using any special hardware, because using hardware always makes problems easier, was because we wanted to make sure that technologically there is no barrier between what we do now and doing the same at home with a phone in rural America or in the middle of Africa or wherever, where you barely have where everything you have is just cellular reception. And the bottom line is that I think overall what we hope is that we are going to be able to give these measurement tools, because what NeuraLight does is just a measurement tool. We don't substitute the thought process or the decision making of anyone. We're just enabling it with surfacing more information. And when physicians don't need to touch and see a patient, by the way, you don't even need to be in rural America. Even if you live in the middle of New York, then if the process of being tested and understanding something, and not in a very good way, about your status means going through these really long, tedious questionnaires that even to go through that, you need to be a awesome Parkinson specialist, for instance. It means that, the amount of people that can get this information is limited, just because how many 1-hour tests can a Parkinson specialist do in a day, and how many Parkinson specialists do you think there are, like literally in the world? So what we want to do is, just like every breakthrough that came by giving more access to information, we want to make sure that instead of using their time and their capability on assessing patients by, these super cool physicians like Professor Getz or Professor Askol, they're amazing at this. They saw thousands of patients. There is a huge craft in what they do. But if instead of spending time on that, they could spend their time on thinking about the patient and giving them the care and the empathy that they need, and will not even need to be right next to them, because the information, you cannot push a patient from far away. But if you have all the information, then using your brains, your insights, and your decision making, you can do that from everywhere. So kind of that's the goal. Not to mention that a lot of the information is probably something that we as healthy individuals would love to have on ourselves. So it's still not happening in our age, but we don't need to get way older than we are until we're going to start thinking, oh, well, is my memory slipping? Am I less sharp than I was? And it's very common for people to be extremely worried or stressed about maybe things going on. And think how nice it could be if you could just easily test whether you're getting better or worse. Maybe you're sleeping more, so things are getting better, or you should stop drinking or whatever. And enabling this access to information in neurology, this is what we want to do, in the sense that just as people take blood pressure tests at home to make these, to have a better feel of where they are, we want them to be able to test themselves in the sense of their cognitive function at home, to have a better feel where they are, and to give better information for their physicians that can actually help them make better choices and get better treatment if they need it.
Absolutely. No, it's exciting. And I think this is an untapped domain. Again, I'll restate it there, because as you were mentioning, we're all, as we all live longer, this is, an area that, maybe 100 years ago we didn't encounter as much, but and I know everyone listening here wants to maintain their faculties, and it's just extremely important, in sort of the healthy aging equation here. So I appreciate you for all you're doing and the way you think about this as well. Eddie, what's coming up? I mean, what else should we be on the lookout for in terms of you, the company, as we get close to 2026? Any major conferences, whether they're on the neurology side, investor conferences, anything else happening that you want to mention? We have you today, please.
So our main focus in the next year or two is, first of all, we want to deepen our partnerships with our pharma partners that spanning all the way from Parkinson's and ALS and the PSP and Huntington's to other diseases as well. It's one of these situations where, the more we manage to help them in earlier stages, hopefully we're going to start seeing more drugs getting further into the funnel, and most hopefully when I need them, then they will already be there. Because it's like one of these things that all of us are going to get old one day. So And on the other hand, just as I said before, we are getting into the area of longevity, and we want to enable giving people information on their cognitive function, not in the sense of when people have Alzheimer's, God forbid, but when people are healthy and they just want to know where they stand. And we're starting a few very exciting collaborations with some longevity clinics.
Nice.
And our goal in the end is, as I said before, to be like the blood pressure test that anyone can take at home. But we're trying to take measured steps on the way there, so that we see that we touch more patients and more lives on the way on one hand, and on the other hand, do meaningful things and make sure that every step is very well validated and with lots of scientific and clinical weight behind it.
Yeah, and that in that whole area of longevity clinics is really coming online the last year or so. So I think you have some major customers there and a whole other sort of patient cohort. So it's exciting. I'm excited for you. I really look forward to seeing you continue to get this technology out there and all, across all these different indications, because they're just, again, so important, many of them unmet, and just really appreciate this work that you're doing. Again, for everybody out there that's going to be listening to the episode across the various podcast networks, or who will be watching on the YouTube channel. Again, you've been spending time with Edmund Benami, co-founder and chief executive officer of NeuraLight, doing really amazing things on both the neurologic drug development side, but also on the patient care front. Eddie, again, I want to thank you for taking the time out of your schedule to come talk to us today for a little while about everything you're up to. Thank you for what you do at the company. And as we like to say on our show, thanks for creating a better tomorrow for so many people via what you've been developing there. Really an awesome story, and I look forward to continuing to follow it.
Thank you so very much. And in the end, people like you that make all of us understand what's important are making a huge impact on all of us.
Really great having you.
Thank you so much.